So in the last video, I discussed up to the process of fertilization and till prevention of polyspermy. So after fertilization has occurred, the genetic information in the gametes, that is both male gamete and female gamete, sperm and egg, they must fuse together and then they must form a diploid cell which is known as the zygote. So here if you can see that the egg has haploid number of chromosomes which is 23 in case of humans. So and sperm also has 23 chromosomes. So here N is equal to 23. And when these two fuse together, their genetic material makes up a total of 46 chromosomes in 23 pairs. These are known as homologous pair of chromosomes. And these 46 chromosomes is the 2n number or diploid number of chromosomes in case of humans. And this is remain conserved throughout generation, and generation after generation the haploid gametes are formed so that the diploid number of chromosomes can be restored in one particular organism. So in case of mouse zygote, it is seen that the two nucleus, these are known as the male pronucleus and the female pronucleus, they undergo the nuclear envelope breakdown. And after the nuclear envelope has broken down, the two spindles are formed separately around each genome. And you can see that these two spindles come closer together and there is a spindle fusion. And after that, during anaphase, one strand of each chromosome goes to one pole and the other strand goes to the other pole. And here you can see that the new nucleus contains the genetic information from both the male gamete as well as the female gamete. And then again the nucleus envelope will reform and the cytokinesis will happen, that is the cytoplasm will divide and it gives rise to two-cell embryo. So from one cell zygote the two-cell embryo is formed, and you can see in these two cells the genetic information is of both the parents. So here the maternal and paternal chromosomes, they assemble their own spindle in this is the mouse zygote that I am talking about. Now there is a controversy regarding this spindle formation, whether the spindle formation is single or double. But now recent studies in the mouse zygote have shown that these spindles are double in case of mice. And after this two-cell embryo is formed, then cleavage will start. So I have already discussed what is cleavage. This cleavage is a series of mitotic divisions where the cell cytoplasm is getting decreasing and the number of nucleus is increasing. So you can see it gives rise to a number of cells in this and the cell size is getting smaller and smaller, because if you see here that this cell after first division the two cells are here. So this is the entire size of the embryo, and after morula stage you will see that the embryo size is remaining the same, but the number of cells inside the embryo is increasing. So cell size decreases, individual cell size decreases, the total size and shape remain the same, cell number increases, and the nucleus to cytoplasm ratio increases. What does that mean? That the number of nuclei will increase and the amount of cytoplasm in each cell is going to decrease, because the amount of cytoplasm is the same and only the nucleus is dividing and giving rise to number of cells. So these are the characteristic features of the cleavage. And after morula stage, another set of division starts and gives rise to the blastocyst stage, which is the hollow wall of cells. As you can see, it has an outer layer of cells which is known as trophoblast, and a cavity which is known as blastocele, and at one end a small cluster of cells which is known as the inner cell mass. And all these individual cells of the blastula or the blastocyst are known as blastomeres. So this is what cleavage is, that is divided. The cytoplasm is divided into numerous small nucleated cells, and these are called blastomeres. So according to the amount and distribution of yolk, eggs can be classified into the following types. So here you can see that according to the basis of the amount of yolk. If yolk is absent, it is known as alecithal egg. If it is present in very little amount, it is known as microlecithal or oligolecithal egg. If it is present in moderate amount, like neither too less not too low, not too large, then it will be known as a mesolecithal. And in case where the egg contains a large amount of yolk, then it is known as a megalecithal, macrolecithal or polylecithal. Same thing, different names are given. And on the basis of distribution of yolk. So if the yolk is distributed uniformly throughout the egg, this is known as homolecithal or isolecithal egg. And if it is located or concentrated only in the central part of the egg, it is known as centrolecithal. And if the yolk is concentrated at one end, entirely at one end of the embryo or egg, it is known as telolecithal. And if it is diffused entirely throughout the egg, then it is known as meiolecithal. So according to that, you can see there are some examples given, like in case of alecithal egg where little or none yolk is present, like no yolk at all or very little. Very little, you can also call it microlecithal or oligolecithal. So in that case, the cleavage, I'll go to the types of cleavage in the next slide. So in that case, blastocyst will be formed, and it is seen in mammals, because in case of humans also, the egg contains very little yolk or no yolk at all. So our egg type is alecithal. In case of centrolecithal, and why the mammal contains eggs with so little yolk? Because the development in case of mammals occurs within the body of the mother only. So since in that case the placenta is responsible for transporting the nutrients and everything, the yolk is not so much required. And in case of centrolecithal egg, like you can see that the centrolecithal has yolk in the central part. So the result will be a blastoderm, means the lining of the egg will only form the blastula, and the center part is covered with yolk. This is seen in case of insects. In case of isolecithal, it is evenly distributed throughout the yolk. Then you will see that it is known as a blastula, and it is seen in sea urchin. Mesolecithal, also the same thing, it is seen in amphibian. And in case of telolecithal, the blastodisc will be formed. Since at entire one end, like here you can see, if the large portion of the egg is covered by yolk and it is concentrated at one end, that is the vegetal pole, then in the animal pole a disc-like blastula will form, because this is the only part where cells are present. Like in the blastula, the blastomeres are only here, located here, and the entire egg is covered with yolk. So in that case, a disc-like blastocyst will form, which is known as a blastodisc. This is seen in case of bird eggs, fish eggs, where the amount of yolk is very high. So this is mesolecithal, like I said, moderate amount of yolk. So you can see almost half of the egg is covered with yolk in the vegetal pole, and the other half has the cell. And this cell will give rise to more and more number of cells, and here the cleavage will occur and blastula will form. And in case of meiolecithal, you can see that little amount of yolk is there, but it is diffused throughout the egg. And in case of alecithal, very little amount of yolk or no yolk at all, and the yolk is not disturbing the cell division in case of the blastula. So these are the types of eggs. Why is it important? Because according to the type of egg, the type of cleavage will be determined. So I'll go to the type of cleavage in the next slide. So before going into the types of cleavage, at first you have to know what are the planes of cleavage. So in case of an egg, this is the animal pole, this is the vegetal pole, and we know that the animal pole contains the nuclear cells and the vegetal pole contains the yolk. So if the axis or the plane of cleavage passes through the polar axis, that is the axis of from the animal pole to the vegetal pole, this plane is known as meridional plane. Like in geography, we have heard about meridian and equator and latitude, longitude. In the same way, the planes are described in case of cleavage. So if the plane runs from animal pole to vegetal pole, this plane right through the center of the egg is known as meridional plane. And if it occurs in the central part of the egg in the equatorial region, it is known as equatorial plane. So the meridional plane and the equatorial plane will be always at right angles to each other. And in case of the planes which are parallel to the meridional plane and perpendicular to the equatorial plane, those planes are known as longitudinal plane. They are parallel to the meridional plane, but they are not passing exactly through the center of the egg. So those are known as longitudinal or vertical planes. And just the reverse, if the planes passing through the other part of the egg other than the center but parallel to the equatorial plane and perpendicular to the meridional plane, then those planes are known as transverse plane or horizontal plane or latitudinal plane. So according to that, different kinds of cleavage have been classified. Like here you can see that the pattern of this cleavage is determined by two major factors. So one is the amount and distribution of yolk. Like I said, that the amount and distribution of yolk within the egg is very important to determine how the cleavage will occur. And the other factors present in the cytoplasm, which are known as molecular cues or signals that influence that at which angle the spindle will move and exactly from where the cell division will start. So this cytoplasmic signals or factors and the yolk are the two parameters which determine how the cleavage will occur in case of an organism. So the major two divisions of cleavage are known as holoblastic cleavage and meroblastic cleavage. So in case of holoblastic cleavage, as you can see that the cleavage is complete. What do I mean by complete? That the division has occurred throughout the embryo and the cells are getting formed, like from 1 cell to 2 cell, 2 cell to 4 cell, 4 to 8. This kind of cleavage is known as holoblastic cleavage, and the entire egg or embryo is divided. So the division is complete here, and it can only happen when the egg contains very little amount of yolk or no yolk at all. So you can see in case of alecythal, the cleavage is holoblastic. Isolecithal, it is holoblastic. Mesolecithal also, it is holoblastic. But in case of telolecithal, telolecithal, it is meroblastic. So what is the meroblastic cleavage? It is the incomplete cleavage which occurs when the egg contains a lot of yolk. So here you can see that this portion is entirely covered by yolk, which can be found in telolecithal eggs or macrolecithal eggs. So here only this part is the cellular part in the animal pole, and this part only can divide by successive mitotic divisions. So the cells are formed only in this portion only, in the blastodisc region. So this kind of cleavage is called incomplete cleavage because the entire embryo or the entire egg cannot be divided. Only the portion where the yolk is not present, that is getting divided and forming a series of cells. So this is known as incomplete or meroblastic cleavage. And in case of here where the yolk content is so low that it does not interfere with the division or the cleavage of the egg and the cleavage is complete, like the division runs from here to here, it is very much complete. In that case, it is known as a holoblastic cleavage. Now, holoblastic and meroplastic both have different types, subtypes according to the plane of cleavage. So, in case of holoblastic cleavage, when the egg is isoleucithal, here you can see that radial cleavage is occurring in echinoderms and amphioxus. So, what is radial cleavage? Like the first plane of cleavage will be meridional, and the next plane will be perpendicular to it. Like this plane, this is the another plane of cleavage is like transverse, and next again from here the cleavage has occurred from the equatorial plane, and it is giving rise to 8 number of cells which are equal in size and shape. So these equal blastomeres, when the cleavage is equal, it divides the embryo into equal size blastomeres at both the poles. In the animal pole also you can see that the 4 blastomeres are of equal size, and the vegetal pole also the 4 blastomeres are of equal size. Then these blastomeres are known as mesomeres. Okay, so one plane will be perpendicular to the other plane. This kind of cleavage is known as radial cleavage. And this next is the spiral cleavage. So in the case of spiral cleavage, as you can see that in the polar axis, the cells will be arranged spirally around the polar axis, and here you can see unequal sized cells have formed. So some blastomeres at the top are smaller in size, they are called micromeres, and the other blastomeres at the bottom, that is the vegetal pole, they are larger in size, which are known as macromeres. So in this kind of cleavage, the cells are situated on top of the furrow. Like in this cell and this cell has a furrow, and in the middle the top cell has formed. So in this case, the cells are exactly on top of one another, but in this case, the cells are arranged on the gaps of the bottom cells. So here in this way, the spiral kind of arrangement will occur, and this kind of cleavage is known as a spiral cleavage. This next is the bilateral cleavage. So here just like bilateral symmetry, the plane of cleavage will give rise to 2 equal blastomeres which are just the mirror images of one another, and this bilateral cleavage is seen in tunicates. So here also almost equal sized blastomeres will be formed. Next is the case of rotational cleavage, which is very important because it is seen in the mammals. In case of rotational cleavage, what happens is that the plane of cleavage, at first there will be an equal division. In the next division, the second division, what will happen is that one of the daughter cells will start dividing meridionally, like the plane of cleavage will be through the meridian, and the other daughter cell will start dividing equatorially. That is, the plane will be through the center, horizontal. So in that way, a different kind of pattern, it will seem that it is kind of rotating around the embryo, and this kind of cleavage is seen in mammals and nematodes. So all these are in case of isoleucithal or aleucithal eggs. Next, when the egg is mesoleucithal, that is moderate amount of yolk is there. So in that case, even if there is holoblastic cleavage, the cell numbers will be unequal because the yolk contained in this pole, in the vegetal pole, will be higher than the animal pole. So animal pole contains small sized blastomeres which are called micromeres, and the large yolk laden blastomeres are called the macromeres at the vegetal pole. This is seen in case of amphibians. Next is the meroblastic cleavage. Like I said, that it is incomplete cleavage. So in all the cases, you can see that the egg is not entirely divided. Here, in case of teloleucithal eggs, that is when the yolk content is concentrated at one end of the egg, then bilateral cleavage, if occurs, it will be like this. One cleavage plane will go like this, the other will go like this, and here successive planes will form cells which are almost equal or mirror images of each other, but the cleavage will be only restricted to this portion, the rest of the egg will have the majority of the yolk, and the cell division will not occur over there. Next is the discoidal cleavage, which is occurring in case of fish, reptiles, and birds. Like I said earlier, that the blastula is called blastodisc here because of the disc-like structure, and the entire yolk content of the egg is restricting the blastula to be formed throughout the egg. The blastula is concentrated in this region only. So in that case, successive divisions will occur here, and this is known as a discoidal cleavage. Next is the centrolecithal egg. In case of centrolecithal egg, like I said earlier, the central part of the egg will be concentrated with yolk. This is seen in case of insects. So at first the nucleus starts dividing. There is no cytoplasmic division, only nucleus division is occurring. And when all the nuclei are arranged to the periphery of the yolk is concentrated at the center, all the nuclei getting arranged around the periphery, and then cell division starts. So in this case, only the periphery of the yolk or the embryo will be divided into cells, and the central part will remain as it is. So this kind of cleavage is known as the centrolecithal superficial cleavage. Superficial cleavage because only the superficial part of the embryo is getting divided, the central part remains unchanged. Next, two other types of cleavage: indeterminate cleavage and determinate cleavage. So if the cells are separated from each other, then each cell has the potential, like each daughter cell can give rise to an entire organism on its own. So this kind of cleavage is seen in case where the regulative development is there. I have already discussed what is regulative development and what is mosaic development. So in this case, even if some cells are missing, each cell has the capability to give rise to an entire organism. This is seen in case of invertebrates such as echinoderms and vertebrates. But in case of determinate cleavage, if the cells are separated, they will die. Like neither of the cell has the potential to give rise to an entire organism on its own. So in that case, all the cells are required together. Only then will the organism be formed. So this is known as mosaic development. I have discussed this earlier, and it is seen in annelid mollusks and ascidians. So next is the extra-embryonic membrane. So the embryo must be protected since it's a very delicate structure. So the embryo is covered by certain membranes which are known as extra-embryonic membranes. So we have 4 extra-embryonic membranes over here, like chorion, amnion, allantois, and yolk sac. All these, they have different specific functions, and they are formed at different locations from the embryo, and they are performing certain functions of their own which are very important in the development of the embryo. So here when you see that this is the embryo, and the first membrane that covers the embryo is the amnion. Inside this amnion is the amniotic cavity, which is filled with the amniotic fluid, and it acts as a very good shock absorber for the embryo so that it is protected from the environment, and it also gives the embryo an aquatic environment to develop, and all the nutrients and everything is passed on, or it is excreting. Whenever it is excreting also, that is also coming out in the amniotic fluid. And this is the yolk sac, which in case of humans is very less developed, but it is there because yolk is very little or no amount at all in case of human embryos. And this yolk sac, in case of other embryos, it is responsible for providing nutrition or food to the growing embryo. And this is known as allantois. This green colored membrane is known as allantois. This allantois is responsible for serving as an excretory organ as well as a respiratory organ. So this allantois can exchange gases with the embryo, O2-CO2 exchange. Also it can collect or store the excretory materials of the embryo and then give back to the mother's body, and from the mother's system it gets excreted outside. So this connection is done through placenta. So I have already discussed what placenta is. When the mother's body and the fetus body forms a connection between them, this connection is made possible by another extraembryonic membrane which is known as chorion. So chorion is the outermost membrane, and it is all obviously giving protection to the embryo, and also it has another function, that is it forms an intimate connection with the mother's body through the placenta. So this is known as the endometrium of the uterus, and you can see that connections are formed, and these connections are known as chorionic villi. So this is the embryonic portion of the placenta, and this is the maternal portion of the placenta, and both together form a intimate connection through which the exchange of nutrients, gases, excretory materials occur from the mother body to the embryo body. And this connection of the allantois, yolk sac and the placenta that you are seeing, this tube is known as umbilical cord. This umbilical cord is the main cord which connects the mother's body with the embryo body. And here if you see that different colors are given to the different membranes. So if this is the embryo, as you can see here, the amnion is here, this one. And from the amnion, another invagination is here, which is known as allantois. This one is allantois. This is the yolk sac, and it occurs from the gut of the embryo. From the gut, this membrane comes out, evagination is occurring, and this portion is the yolk sac, which contains the yolk, and this is the allantois. And the embryo is covered by the membrane known as amnion. This is the amniotic cavity, and the entire thing is covered by another membrane which is known as chorion. So in case of amnion and chorion, you will see that the colors are same, like blue and red, blue and red. So here red is the endoderm and blue is the ectoderm. So with ectoderm and mesoderm, amnion and chorion are formed, and this ectoderm plus mesoderm is known as somatopleure. And in case of yolk sac and allantois, endoderm and mesoderm are taking part, and this is known as planchnopleure. So ectoderm plus mesoderm is known as somatopleure, and endoderm and mesoderm is known as planchnopleure.